We live surrounded by things that seem ordinary and familiar, but when we zoom in on the world under a microscope, these everyday objects appear quite bizarre. So today, I'll be exploring 10 of the strangest things you can see under a microscope. Snowflakes are often described as thin or symmetrical, but their true complexity is only revealed under a microscope. A single snowflake is typically between 1 and 5 millimeters in diameter. However, the intricate details of their structure exist on scales thousands of times smaller. The light microscope and the electron microscope have been used for decades to document how snowflakes form and why no two are exactly alike. At the microscopic level, it's just a single ice crystal. Each crystal begins to form around a tiny nucleus, usually a dust particle, pollen grain, or other microscopic particles suspended in the atmosphere. When water vapor freezes on this nucleus, it arranges itself into a hexagonal crystal. This hexagonal symmetry isn't arbitrary either. It's determined by the molecular structure of the water itself. Upon magnification, it reveals branching arms, edges, and terraces growing outward from the center. These structures form when the crystal experiences varying conditions of temperature and humidity as it falls through the atmosphere. This microscopy has shown that even slight changes in these conditions can alter growth patterns. This is why snowflakes exhibit a wide range of shapes, from thin sheets to needle-like forms to complex tree-like branches. One of the most important insights the microscope provided is that the symmetry of a snowflake is approximate, not perfect. While the arms of a snowflake often appear identical to the naked eye, microscopic images reveal subtle differences between them. Each arm is exposed to slightly different conditions as the snowflake falls, resulting in variations. This explains why the common phrase, perfectly symmetrical snowflake, is not scientifically accurate. The microscope also revealed internal structures within snowflakes. Cross-sectional images show that snowflakes often contain air pockets and stratified growth areas. These internal features affect how light is reflected on a snowflake, causing that white appearance of snow when many crystals are stacked together.
Household dust. Household dust appears harmless to our naked eyes, but under a microscope, it reveals a complex ecosystem that's dominated by dust mites. Dust mites are microscopic arachnids measuring about 200 to 300 micrometers in length. They're invisible without magnification and are commonly found in bedding, carpets, upholstered furniture, and clothing fibers. Under microscopy, dust mites appear translucent and vaguely insect-like. Under scanning electron microscopes, they become unmistakable. The images reveal 8-jointed legs, claw-like appendages, and segmented bodies that are covered in fine hairs and ridges. Their mouthparts are specialized for consuming organic debris, primarily flakes of human skin, which humans shed constantly. Microscopes show that dust mites lack eyes and respiratory lungs. Instead, they absorb oxygen directly through their exoskeleton. Their bodies are covered in grooves that increase the surface area, which aids in that gas exchange. And this kind of simplicity allows them to survive in low-oxygen environments like mattresses and pillows. Under the microscope, dust mite fecal pellets appear as tiny oval-shaped particles that are coated with proteins. Those proteins are among the most common indoor allergens worldwide. And microscopy has confirmed that these particles are small enough to become airborne and inhaled, where they can trigger immune responses in sensitive people. When household dust is examined under a microscope, it becomes clear that it's not just simply dirt. It's a mixture of fibers from clothing, paper, pet dander, pollen grains, fungal spores, and fragmented insect parts. Each component has a distinct appearance. Textile fibers appear like twisted ribbons or smooth cylinders, depending on their origin. Pollen grains have rigid spike shells. Fungal spores vary in shape but are often spherical or elongated. And dust mites, they thrive in humid environments, and microscopic studies have shown that they're most abundant in bedding because mattresses provide stable temperatures, moisture from human perspiration, and a constant food source. But contrary to popular belief, they don't bite humans and they don't live on your body. Their impact is entirely indirect through allergens. Microscopes have also been used to study the mite population density. In heavily colonized environments, dozens of mites might be found in a single gram of dust. These images explain why allergen exposure can be so difficult to eliminate completely, even in a clean home. Seeing dust mites under a microscope fundamentally changes how household dust is understood. What appears inert and lifeless at a human scale is, at microscopic scale, a living system that's shaped by human behavior and environment. These images aren't exaggerated, they're simply revealing what is already there, present and unseen. Microscopy turns everyday dust into evidence of constant interaction between humans and their surroundings. It shows that even the most ordinary environments contain complex life when viewed at the right scale, of course.
Human blood. Human red blood cells. They're one of the most familiar components of our bodies, yet their true structure is only revealed under powerful microscopes. Each red blood cell measures about 7 to 8 micrometers in diameter. Under a standard light microscope, they appear as faint, pale discs. Under a scanning electron microscope, though, they look dramatically different. Smooth, flexible structures that are shaped like shallow donuts rather than flat circles. This biconcave shape is one of the defining features of red blood cells. Electron microscopy shows that the center of each cell is thinner than the edges, creating an indentation on both sides. This geometry is critical for its function. It increases the surface area relative to its volume, allowing oxygen and carbon dioxide to diffuse efficiently across the cell membrane. The shape also allows red blood cells to deform without breaking as they are squeezed through the capillaries, narrower than the cell itself. Microscopy reveals that red blood cells lack many features found in other cells. They've got no nucleus, no mitochondria, and no internal organs. This absence is not a defect, it's an adaptation. Without a nucleus, more internal space is available for hemoglobin, the iron-containing protein responsible for oxygen transport. Under high magnification, the surface of the blood cell appears smooth and uniform, and it's supported by a complex protein scaffold beneath the membrane that maintains that flexibility. When red blood cells are imaged under abnormal conditions, microscopy can reveal some amazing changes. In cases of dehydration, the cells may appear shriveled or spiked, a condition known as crenation. In certain genetic disorders such as sickle cell disease, electron microscopy can show elongated, rigid cells rather than smooth discs, and those changes explain why such cells can block blood vessels and cause pain or tissue damage. Microscopy has also been used to study how the red blood cells interact with pathogens. For example, images of malaria-infected red blood cells show the surface distortions and knob-like protrusions caused by the parasite Plasmodium falciparum. These protrusions help infected cells adhere to blood vessel walls, preventing them from being filtered out by the spleen. Another surprising observation under the microscope is how densely packed red blood cells become in circulation. Electron micrographs of blood smears show cells pressed tightly together, deforming slightly at the contact points. That confirms that red blood cells are not rigid objects, but dynamic structures that are constantly changing shape as they flow. Each cell is essentially a membrane sac filled with hemoglobin optimized over millions of years of evolution. And microscopy doesn't exaggerate this complexity. It reveals how a simple-looking cell is precisely engineered for survival. Seen up close, red blood cells look less like biological tissue and more like an engineered component. Their smooth geometry makes them one of the clearest examples of how form follows function at the microscopic level.
Demodex mites. Boy, they got a good name. Demodex mites. They're microscopic arthropods that live on human skin, primarily within hair follicles and sebaceous glands. Two species are known to inhabit humans, Demodex follicularum and Demodex brevis.
These mites are typically between 0.1 and 4 millimeters long, making them invisible without magnification. Under a microscope, Demodex mites have an elongated worm-like body with 8 short legs clustered near the head. Their bodies are segmented and semi-transparent, which allows the internal organs to be seen under certain imaging conditions. Scanning electron microscopy reveals a rigid exoskeleton with fine ridges and scale-like textures along the body. Observation shows that Demodex mites are anatomically specialized for life inside hair follicles. Their legs are short and adapted for gripping the follicle walls rather than walking on open surfaces. Their mouth parts are designed to feed on sebum, dead skin cells, and cellular debris rather than blood. And they don't bite or sting, which is nice. Demodex mites are most commonly found on the face, particularly around the nose, the eyebrows, eyelashes, and forehead. Microscopy has confirmed that almost all adult humans host the mites, often without any symptoms. Studies using skin scrapings and follicle samples showed that mite populations increase with age, likely due to changes in skin oil production. Under high magnification, researchers have observed Demodex mites positioned head down inside follicles, feeding and laying eggs. The eggs hatch into larvae, which develop through several stages before reaching adulthood. The entire life cycle happens on your skin and typically lasts only a few weeks. Microscopy has confirmed that Demodex mites lack an anus, meaning waste accumulates inside their bodies and is released only when the mite dies. In most cases, Demodex mites cause no harm. However, microscopy has been used to study their role in certain skin conditions. Elevated mite densities have been associated with certain inflammatory skin disorders. And while the exact causal relationships are still being studied, microscopy provides direct evidence of increased populations in those affected areas. But seeing the Demodex mites under a microscope is unsettling for many people, me included. Not because they're dangerous, but because they reveal a hidden reality of our biology. These organisms are not invaders in the traditional sense. They're long-term residents that have coexisted with humans for thousands of years. Microscopy doesn't sensationalize these mites. It simply makes visible a biological relationship that has already existed. At the microscopic level, human skin is not a sterile surface. It's a complex habitat, and Demodex mites are part of that system.
Tardigrades. Commonly known as water bears, tardigrades are among the most studied microscopic organisms because of how extreme their survival abilities are. They typically measure between 0.1 and 1 millimeter in length, which places them right at the edge of visibility to the naked eye. Under a microscope, though, their appearance is unmistakable and surprisingly complex. They've got plump, segmented bodies, 4 pairs of stubby legs, and claw-like structures at the end of each limb that they use to grip surfaces. Under light microscopy, tardigrades appear translucent, allowing some internal structures to be seen. Under electron microscopy, their outer cuticle becomes more apparent. This cuticle is layered and flexible, which allows them to expand and contract as they move. Their mouths contain a specialized feeding apparatus with a stylet-like structure used to pierce plant cells, algae, or small invertebrates and extract nutrients. Now, what makes them remarkable is their ability to survive conditions that are lethal to almost all other known forms of life. Under the microscope, researchers have observed tardigrades entering a state called cryptobiosis. In this state, the organism expels nearly all the water from its body and curls into a compact, dehydrated form known as a ton. Microscopy confirms that during this state, metabolic activity drops to levels that are nearly undetectable. Their cell membranes and proteins are stabilized by special molecules, including unique proteins that replace water and help prevent any structural collapse. These proteins have been identified in studies and laboratory conditions, and when rehydrated, tardigrades can return to active life even after years in a dormant state. Imaging has also documented the tardigrades surviving extreme temperatures from well below freezing to well above boiling, as well as intense radiation exposure and even a vacuum condition. Images taken after such experiments show intact tissues and organs, which show that their survival is not due to avoidance of damage, but to cellular-level protection and repair. Under a microscope, tardigrades also reveal a fully developed internal system. They've got a digestive tract, muscles, nervous system, and reproductive organs. They're visible through staining techniques and high magnification imaging. But despite their size, they're not simple organisms. Their anatomical complexity rivals that of much larger animals scaled down to a microscopic level. Seeing a tardigrade under a microscope does often change how people think about life at small scales. They don't look fragile or primitive. Instead, they appear robust and purpose-built, with structures that explain their durability rather than exaggerated. Every survival claim about tardigrades have been verified through repeated observation and controlled experiments. And under magnification, they're not monsters or curiosities. They are complete animals, operating at a scale where survival requires this extremity. Microscopy simply reveals what evolution has already engineered.
Diatoms. Diatoms are single-celled algae that possess some of the most intricate structures ever documented. Measuring between 2 to 200 micrometers in size, diatoms are found in nearly every aquatic environment on the planet, including oceans, freshwater lakes, rivers, and even damp soil. The defining feature of them is the cell wall, known as the frust. The frust is composed primarily of silica, the same kind of material found in glass. Electron microscopy reveals that each frust consists of two interlocking halves, similar to a petri dish, with one half fitting inside the other. These halves are covered in pores, ridges, and patterns arranged with amazing precision. The patterns observed in diatom frustals are not random either. Microscopy shows that pore size, spacing, and arrangement are species-specific, and they're functionally significant. Some diatom species have pores measured in nanometers, placing them at the boundary between biological and nanostructured materials. Thousands of species have been identified based solely on the frustal morphology. This kind of diversity of shapes includes circles, triangles, stars, rods, and even elongated forms. Each shape corresponds to specific ecological adaptations, like buoyancy control or attachment to a surface. But let's back up a bit. They play a critical role in the Earth's ecosystems. Diatoms are responsible for a significant portion of global oxygen production through photosynthesis. They form the base of many aquatic food webs, supporting organisms ranging from microscopic zooplankton to large marine animals. Some species lack flagella yet are capable of some gliding motion. Imaging shows that this movement is facilitated by the secretion of a substance through specialized structures in the frust, and this allows the diatoms to slowly reposition themselves on a surface. Over time, though, massive accumulations of fossilized frustals form diatomaceous earth, a naturally occurring sediment used for filtration and insulation. Electron microscopy has confirmed that it's composed almost entirely of intact frustal fragments. These diatoms have attracted interest from materials scientists, too. These investigations rely heavily on electron microscopy to analyze frustal geometry at extremely fine scales. Importantly, the research doesn't involve modifying diatoms themselves, but studying the structures that already exist. These naturally occurring nanostructures are studied for potential applications in photonics and even filtration. But when appeared under high magnification, they appear artificial, as if designed rather than grown. This impression is the result of scale. Their symmetry and precision are natural outcomes of a biological process operating at these microscopic levels. Under the microscope, diatoms are clear evidence that some of the most elaborate structures on the planet exist at scales humans rarely even notice.
Pollen grains. Pollen grains are among the most visually diverse structures ever observed under a microscope. Although they're commonly associated with allergies and plant reproduction, their true complexity only becomes apparent under magnification. Most pollen grains measure between 10 to 100 micrometers in diameter, and they're well beyond the resolution of the human eye, but easily within the range of light and electron microscopy. Under scanning electron microscopy, however, they reveal a highly detailed outer shell known as an exine. These shells are made primarily of sporopolyen. It's one of the most chemically resistant biological materials that's known. The durability of it is so extreme that pollen grains can remain intact in sediments for millions of years, allowing scientists to reconstruct ancient climates and vegetation. Now, the microscope shows pollen egg scenes. They're not smooth. Instead, they're covered in ridges, spikes, pores, lattices, or spines, depending on the plant species. Each species produces pollen with a distinctive surface pattern. And they're not decorative. They do serve a functional purpose, like aiding an attachment to the pollinator or influencing how it interacts with the air. For example, pollen grains from wind-pollinated plants like grasses tend to be smooth and lightweight, optimized for airborne travel. In contrast, pollen from insect-pollinated plants often has spines or sticky coatings that help it adhere to an insect. The pollen grain isn't merely a shell. Inside reveals a complex internal structure containing the male gametophyte of the plant. This includes generative cells and tube cells, which are responsible for fertilization once the pollen grain reaches a compatible stigma. In allergy research, microscopy has been essential for understanding how pollen fragments break apart. Certain pollen grains rupture under humid conditions, which release submicron particles small enough to penetrate deep into your lungs. And electron microscopy has confirmed the size and structure of these fragments, helping explain why thunderstorms can trigger severe asthma events in some people. Each species of pollen produces grains that are nearly identical in shape and structure, generation after generation. This reliability allows scientists who study pollen to identify plant species based solely on the pollen samples. Microscopy doesn't turn pollen into something strange or alien. It reveals that even the smallest components of plant reproduction are built with extraordinary complexity. They're hidden entirely from everyday perception.
Bacteriophages. Bacteriophages, often shortened to just phages, are viruses that infect bacteria. They're among the most abundant biological entities on Earth, with estimates suggesting there's more than 10 to the 31st power phage particles globally. Despite the huge numbers, though, they're completely invisible without a powerful microscope. Most range from about 20 to 200 nanometers in size, and they're well below the resolution of light microscopes and firmly within the domain of electron microscopy. Many of them have a distinct head and tail structure. The head, known as the capsid, is typically icosahedral in shape and contains the viral genetic material, either DNA or RNA. Attached to the head is a tail structure that varies in length depending on the phage type, and some tails are short and rigid, while others are long and contractile. Microscopy shows that the tail isn't decorative. It functions as a molecular injection system. At the end of the tail are fibers or spikes that recognize and bind to specific receptors on a bacterial cell. These receptors are often proteins or polysaccharides that are unique to bacterial species, and that specificity explains why most bacterial phages can infect only particular types of bacteria. Once attached, high-speed electron microscopy studies contractile-tailed phages that physically change shape. The tail sheath will contract, driving an inner tube through the bacterial cell wall and membrane that allows the viral genome to be injected directly into the bacterial cytoplasm. The phage itself remains outside the wall, only the genetic material enters. Under electron microscopes, researchers have captured images of bacteria in various stages of phage infection. Some images show bacterial cells packed with newly formed phages, while others show ruptured cells surrounded by released viral particles.
This provides direct visual confirmation of the process, first inferred through biochemical experiments. But not all of the bacteriophages immediately destroy their hosts. Some enter a dormant state. In these cases, microscopy alone can't distinguish dormant phages, but genetic and structural studies confirm that the phage genome integrates into the bacterial chromosome. The phage can remain inactive for many generations, replicating alongside the bacterial DNA until it conditions trigger activation. Microscopic images of seawater samples consistently reveal huge numbers of phages interacting with maritime bacteria. The interactions influence their nutrient cycles and ecosystem stability. And in oceans alone, phages are responsible for killing a significant fraction of bacteria every day, recycling the organic material back into the environment. In medicine, microscopy has been essential for renewed interest in phage therapy. Electron micrographs are used to verify phage structure, purity, and interaction with the target bacteria. And while phage therapy is still under controlled development in many countries, the physical behavior of phages observed under microscopes is reproducible. They look less like biological entities and more like a microscopic machine, complete with an injection system, an anchoring structure, and even symmetrical components. Their function depends on these physical structures, and microscopy doesn't dramatize these bacteriophages, it reveals them as they are. They're highly specialized biological systems operating at nanometer scales, shaping bacterial life and death across the planet.
Human hair and skin. Human hair and skin appear smooth and uniform at everyday scale, but under a microscope, they reveal complex structures shaped by biology, environment, and the wear. Human hair typically ranges from 17 to over 180 micrometers in diameter, depending on genetics and location on the body. Skin cells are even smaller, forming layered structures that are constantly renewed throughout life. A strand of hair appears as a simple filament under scanning electron microscopy. However, its surface becomes highly detailed. Hair shafts are covered in overlapping scales made of keratin, forming a structure known as the cuticle. These scales lie flat in healthy hair, creating a smooth surface, and microscopy shows that damage from heat, chemicals, or friction cause the scales to lift, crack, or break away. The internal structure of hair is also visible under higher magnification. Beneath the cuticle lies the cortex, which contains the tightly packed keratin fibers and pigment granules. These pigment granules determine your hair color, and microscopy has shown that differences in pigment size, shape, and distribution do correspond directly to variations in hair color and texture. Human skin, on the other hand, reveals an entirely different level of complexity. The outermost layer, the stratum corneum, consists of flattened, dead cells packed tightly together. Under magnification, these cells resemble overlapping plates rather than smooth tissue, and this layer acts as a physical barrier, and it prevents water loss and blocks pathogens. Beneath the surface, though, microscopy reveals living cells in various stages of division and differentiation. Skin cells migrate upwards over the weeks, gradually flattening and losing their nuclei before forming the protective outer layer. This process is directly observable using histological staining and some microscopic imaging. Skin from the palms and soles of the feet have a much thicker outer layer, while facial skin is thinner and more sensitive. Pores, hair follicles, and even sweat glands appear as a complex opening and channel under magnification, each with distinct structures and functions. One really neat feature is the presence of micro cracks and abrasions on everyday skin. Even skin that appears intact to our naked eye often show fine fractures under magnification. These microstructures explain why skin can absorb chemicals and microorganisms more easily than expected, a finding supported by decades of dermatological research. Microscopy has been used to study aging, too. Under magnification, aging skin does show changes in collagen fiber density and organization, while hair shows thinning cuticles and increased surface irregularities. These changes correlate with known aging processes rather than any cosmetic appearance alone.
Environmental exposure, hygiene practices, and lifestyle all leave measurable traces. They're not subjective interpretations, they're observable changes documented repeatedly in controlled studies. Microscopy doesn't exaggerate their complexity. It simply reveals the physical reality of living tissue at scales humans rarely even seek.
Neurons and synapses. Neurons are the fundamental signaling unit of our nervous systems, and their true complexity is only visible under high-powered microscopes. While the human brain contains about 86 billion neurons, each neuron operates at scales way below what can be seen without magnification. Neuronal cell bodies typically measure around 10 to 50 micrometers in diameter, but the most critical structures, the synapses, are often less than 1 micrometer wide. Under light microscopy, neurons appear like branching cells with long extensions. Under electron microscopy, however, their architecture becomes far more detailed and precise. Each neuron consists of a cell body, dendrites that receive signals, and an axon that transmits signals. The axon can extend from millimeters or even over a meter in the human body, yet it maintains a diameter measured in micrometers. Microscopy has revealed how neurons don't physically touch each other in most cases. Instead, they communicate across tiny gaps called synaptic cliffs. These gaps typically measure about 20 to 40 nanometers across, and that distance is so small that thousands of synapses could fit across the width of a human hair. At a synapse, microscopy has showed an organized arrangement. On the transmitting side, known as the presynaptic terminal, there are clusters of synaptic vesicles filled with a neurotransmitter molecule. These vesicles are typically about 40 to 50 nanometers in diameter, and they're positioned near the presynaptic membrane, ready to release their contents. When a neuron fires, electrical signals trigger calcium ions to enter the presynaptic terminal. Electron microscopy combined with biochemical studies has confirmed that this calcium influx causes vesicles to fuse with the membrane and then release neurotransmitters into the synaptic cliff. These molecules diffuse across the gap, and then they bind to receptors on the receiving neuron. The receiving side, called the postsynaptic density, appears under the microscope as a thickened region packed with receptors and scaffolding proteins. The density is not uniform. The size and shape vary depending on the strength and type of the synapse, but stronger synapses often have larger postsynaptic densities, which is a relationship confirmed through repeated imaging studies. Neurons are supported by other cell types, including astrocytes and microglia, which are also visible under magnification. Astrocytes extend this fine process that wrap around the synapse, regulating the neurotransmitter levels and providing metabolic support. Microglia act as immune cells, surveying the environment and responding to injury or disease. Microscopy has also revealed how neurons are affected by disease. In neurodegenerative conditions, electron microscopy shows loss of synaptic connections, accumulation of abnormal protein aggregates, and damage to cellular organelles. These changes do correlate directly with cognitive and motor symptoms that are observed clinically. But what makes them cool is how much information transfer happens across these tiny spaces. Every thought, movement, and sensation you have depends on countless synaptic events happening simultaneously across nanometer-scale gaps. This measurable physical process is observed repeatedly through microscopy, and at everyday scale, the brain is intangible. Under the microscope, though,